Well, good morning, everyone. My name is Lori Arrow, and I'm with Pacific Biosciences, otherwise known as PacBio. And I'd like to welcome everyone to the Discovery Life Sciences PacBio webinar on transformation drug research and development via accurate and scalable long-read sequencing. It is my pleasure this morning to moderate our webinar, and I'd like to first just introduce you to our two speakers today. The first speaker will be Cheryl Heiner, and Cheryl Heiner is one of the principal scientists at PacBio. She has interests in metagenomics, microbial, and viral applications, and she has extensive involvement in the single molecule sequencing development at PacBio. Our second speaker today is going to be Shawn Levy, Chief Scientific Officer of Discovery Life Sciences. He is also a faculty investigator at the HudsonAlpha Institute for Biotechnology in Huntsville, Alabama. Dr. Levy received his PhD in biochemistry from Emory University, and his research interests in genomics include the application of genomic technology to rare disease, adult and pediatric cancer, and technology and methods development in high-performance sequencing for gene expression profiling, genotyping, and structural and functional genomics. Other research areas of interest include human performance and physiology and the application of technology to better understand athletic performance, recovery, and disease. Our agenda today is Cheryl will take a moment to introduce the PacBio technology, our highly accurate long-read HiFi sequencing, and she will cover several use case examples of the technology, including our work on SARS-CoV-2, recombinant AAV, gene editing, and our CRISPRCas9 editing, and finally talking a little about our Iso-Seq or RNA transcript sequencing. We will then transition to Shawn, who will cover the scalable applications for drug and diagnostic development programs at HudsonAlpha Discovery, where he will also highlight the All of Us long-read sequencing project and cover expanded detection of large structural variations in population genomic investigations, including oncology and other indications, and the precise characterization of low complexity regions, trinucleotides, and repeat expansions. And at the end, we will have a question and answer period. So a couple of housekeeping notes. All attendees are in muted mode, and questions can be submitted in the chat box at any time, and these questions will be answered at the end. We have a group of folks in the background that will be helping to answer our questions from both Discovery Life Sciences as well as PacBio. The session is being recorded and will be made available to all participants. And we will offer a brief survey to all attendees at the end in order for you to provide feedback. So at this point, I'm going to transition over to Cheryl Heiner, and she will take it from here and begin the presentation on the PacBio system.
Good morning, everyone. I'm excited to be with you today to talk about transformative drug research and development via accurate and scalable long-read sequencing. And as Lori mentioned, I'm from PacBio, representing the PacBio team. So first, here are some key application areas where PacBio really excels, and we'll be discussing many of them today. Some background on PacBio sequencing. We call it SMART sequencing, and SMART stands for single molecule real-time sequencing. And those are really the key features here, which I've listed also below. So we have long reads, they can be tens of KB long, and high accuracy, and we're free of systematic errors, no systematic errors in our sequencing. Additionally, another key feature is that we have single molecule resolution. So we're sequencing single molecules. And then in addition to that, we have uniform coverage independent of the sequence context of your sample. And that's because we have no DNA amplification, no GC bias or PCR artifacts, and our polymerase does not have a problem with any sequence context. And then finally, if desired, you can also get simultaneous epigenetic detection from your samples, assuming you're starting with native DNA. So you can really sequence any type of sample that you like, as long as you can convert it into double-stranded DNA. And here's our smart sequencing workflow. So starting with double-stranded DNA, the first step is to ligate hairpin adapters to both ends of that of all the DNA molecules. And that's what forms what we call our SMRTbell libraries. Then we anneal primer and bind polymerase to the hairpin single-stranded region of those SMRTbells and load them onto our Sequel system. So inside the Sequel system, those samples are then transferred to our SMRT cells, and the SMRT cells contain millions of what we call zero-mode waveguides, our little tunnels. And in a single zero-mode waveguide, one SMRTbell molecule would be immobilized with the polymerase at the bottom of that ZMW. And that's how we're able to get simultaneous real-time detection of millions of molecules sequencing at the same time. Now, how do we get such high accuracy from single molecules? Well, there are two key features here. One is that SMRTbell template structure, and the other is the fact that we're using a strand displacing polymerase. So in the sequencing, the polymerase extends through that double-stranded insert region, opening up our SMRTbells into a circular molecule. And the polymerase makes multiple passes on that circle. So it sequences the insert in both orientations multiple times. Now, the single pass accuracy is not all that high. So as you can see here, something like 90% accuracy with one pass. But because those errors are random, they quickly wash out with coverage. So by the time we're at 3 passes, we're up to 98% accuracy. And then with 10 passes, we have a HiFi read, what we call HiFi, which has 99.9% accuracy or greater. And so this really opens up a new paradigm in DNA sequencing. In the past, you've had to choose. If you want high accuracy, you need short reads. And if you want long reads, then the accuracy is not going to be that high. But with our HiFi reads, you can have both. You can have long reads as well as high accuracy, all in the same sequence. So here's some statistics, numbers from what you can get from one SMRT cell with our latest Sequel 2E system. And this is with the human genome library. The average insert size is about 16 kb from a shared genomic sample. And from one cell, one SMRT cell, we're getting over 2 million HiFi reads, averaging that length, 34 gigabases of HiFi data. And the median accuracy is greater than 99.9%, or Q32. Now, with a 20-fold human genome coverage sample, assembly occurs in the 2E system in approximately 5.5 hours. So it's a lot faster than it used to be. Now you might be wondering, what does a 16 kb read with a Q32 accuracy look like? What's the value of that? So here I have an example for you. And in fact, this is a 19,820 base pair HiFi read with a predicted QV of Q33. In fact, there are only 8 errors in this entire sequence. And they're indicated with the red triangles. So we have 19,812 correct bases for an actual QV of 34. And you can imagine, since these errors are random, it doesn't take many reads of this quality to get an extremely high consensus sequence in this region. So what can you do with HiFi reads? Well, here's a number of applications that I think might be of interest to you. Platinum level, very high accuracy de novo assembly, detection of all variant types, so that would be SNPs, indels, and structural rearrangements with high precision and recall. And then you can phase those variants, because we have long reads, phase into long haplotypes. additional applications, sequencing full-length transcript, and then also sequencing metagenomes and viral populations in high resolution. So I'm going to start now with that last application area, viral populations, with a very important virus today, SARS-CoV-2, full viral genome sequencing. So I would say that actually PacBio HiFi sequencing is the best method for getting complete genomes of SARS-CoV-2. And that's because we can use long amplicons. And here we're using 1.2 kb amplicons that have the advantages shown here. Better primer balancing, just because there are fewer PCR reactions, fewer dropouts because the primers are covering a smaller area of the viral genome, so there's less chance of viral mutations that will cause dropouts. And then because the amplicons are longer, we're able to call all variants. So we're not only SNPs, but also insertions and deletions of up to 300 base pair with only 4-fold coverage. So here's what our workflow looks like, starting with the nearly 30 kb SARS-CoV-2 genome. It's divided into 29 amplicons. So that's an RNA virus. You do RT and then divided into 29 amplicons. Again, 1.2 kb in length. The PCR occurs in 2 pools. Then Smart Bell library is constructed, sequencing. That part of the process takes about 15 hours. The hands-on lab work is about 5 hours. Sequencing about 15 hours. And the entire process can be, or the sequencing can be multiplexed at a level of greater than 900 samples per SMRT Cell. So here's some examples of results that we've gotten in collaboration with LabCorp with our SARS-CoV-2 HiFi sequencing. On the top, you can see an example of a 300 base pair deletion. This deletion is entirely within one of those 1.2 kb amplicons, so it's extremely easy to see. It's just in the data, whereas it would be split between 2 amplicons if you were using the Arctic 400 base pair amplicon set. And then in the bottom, you see an example where there are actually 6 variants, all within one of these 1.2 kb amplicons. They're very cleanly, easily phased into 2 subtypes here. And that phasing, again, would be impossible to do with the shorter amplicons. So now moving on to another viral application that I think will be of interest to you. This is recombinant adeno-associated virus, and here we're using Smart Bell, our Smart HiFi sequencing for QC. So I think most of you are probably familiar with the AAV genome structure. It's a single-stranded DNA virus, has 2 viral genes surrounded by inverted tandem repeats on both ends. And both of those viral genes can be replaced with the gene of your choice up to something like 4.5 kb. When that occurs, in order to get replication, that viral vector plasmid needs to be accompanied by a packaging plasmid and a helper plasmid. So HiFi reads are really perfect for QCing a recombinant AAV library because we get single molecule reads covering the entire AAV genome in one read at high accuracy. And that's exactly what this group from University of Massachusetts Medical School did. And they had some surprising results as described here. So not only did they find the gene that they were targeting in their recombinant vector sequences, but they also found sequences with plasmid backbones. And then additionally, some chimeric molecules, including human sequences. And as you can see in the example on the lower right, here's one molecule that actually had 6 different insertions, including 2 regions of 2 different regions of human DNA, some capsid sequences, some vector sequences. And again, this would have been impossible to put together with short reads. So another application I think will be of interest to you. So this is looking, again, another validation application, but looking at CRISPR-Cas9 editing of genes in the human genome. And so now we're looking at a a study that was done by a group at the Sanger Center, and they're editing the PIG-A locus and with CRISPR-Cas9. And then again, they wanted to use PacBio HiFi reads to QC the results of that gene editing. And so they created a 5.5 kb amplicon around that gene editing site. The gene editing site is indicated by the vertical lines and the red arrows on the left. And then they sorted cells by PIG-A expression. And so let's focus on the blue region in the middle. So these are the PIG-A negative expression cells where you would expect the editing occurred, and there was deletion in that of the gene in that region, which is what you see when you look at the read depth. But then they found some surprising results when they looked at the sequences that were there. So in addition, and that's shown on the right, so in addition to the deletions, they also saw that there were insertions, inversions, and complex rearrangements that occurred. Insertions of the region that was excised as well as other regions. And this was totally unexpected. And again, this could only be seen with this high accuracy single molecule long read resolution. And so finally, another application area that I think is very important, full-length transcript sequencing. So now comparing PacBio sequence to traditional RNA sequencing, the traditional RNA sequencing shown on the top, each transcript is covered by many short reads. So we have fragmented coverage, and putting those reads together to get a full-length transcript is really a big assembly problem. You need reads that span the splice junctions, and even then it's very challenging to, or maybe impossible to figure out what those full-length transcripts look like. In contrast, on the bottom with PacBio Iso-Seq method, it's very easy. Each read represents a full-length transcript. There's no assembly required. So you simply look at the reads and this from a particular sample, and that will tell you the isoforms that were present in that sample. And the splice junctions, it all is just right there. So some important application areas for Iso-Seq, I think for you perhaps, first on top, genetic insights for rare diseases. So this is a work done by a group at Stanford. And what they found, they're looking at transcripts from a previously uncharacterized mutation in a person that had a rare disease, severe hypertrophic cardiomyopathy. And they found that novel alternatively spliced transcripts linked to that mutation, which was new information and provided insight into possible disease cause. And then the second application shown on the bottom, fusion transcripts in cancer. And here we have work from a group at St. Jude's Hospital and studying B-cell ALL. And what they found was the translocation of IGH-DUX4 occurs exclusively into the silenced IGH allele. And this was also an unexpected finding. Both of these types of results are only available with long read, highly accurate sequences that can put these things together. So in conclusion, with the advances that have been, that we've made in terms of throughput, accuracy, ease of use, workflow, there are now many applications that can be done with 1 or 2 smart cells. So very affordable. And here's a list of many of them. I've talked about some of them. Shawn will talk about some others. A few of these we haven't covered, but any of these that are of interest to you, please don't hesitate to contact Shawn or myself. And I think I'll wrap it up there. Thank you very much for your attention.
Thank you, Cheryl. That was terrific. So we will now transition over to Shawn, and he will talk about the capabilities of the Discovery Life Sciences. So Shawn, I will take myself off live, and it is all you.
Great. Thanks. It's a privilege certainly to be here to present some of the work we've done at Discovery Life Sciences and HudsonAlpha Institute for Biotechnology using the PacBio platform over the last, I guess it's been 18 months or so. We've been steadily adding capacity over that time in support of a few different projects. And so I'm going to be speaking about 4 general areas. I will start off with an acknowledgement and mention some of those areas in general. So the main lab team is shown here from the HudsonAlpha Discovery team. I've highlighted in bold some of the key folks that have participated in this long read application and related approaches. And so certainly I'm speaking on behalf of a very dedicated and exceptional team. In addition to that, we have some key collaborators, and I've mentioned them in the slides as we go through them, that are primarily part of the All of Us research program long read working group. There are some individuals from University of Washington, Pete Adano, William Harvey, and Evan Eichler. At Baylor University, Fritz and Medhat, I'll talk about some of their work. And then finally, Kieran Garamella at the Broad Institute and the Data Sciences platform has really done some amazing work on analysis approaches for some of the very unique resolutions that these long read approaches bring. And I'll mention that. In the interest of time, we have some great collaborators at St. Jude Children's Research Hospital. We've started to use the long read sequencing as part of the Gabriella Miller Kids First research program, which we're one of the national genome centers in collaboration with St. Jude. Unfortunately, just due to time today, I won't have too much time to delve into that work, but I think I did want to take a moment to at least recognize, St. Jude has also begun using these applications. And 2 key things I would mention is there's a great 2018 paper from Ma, Liu, and Liu that's published in Nature analyzing 1,699 leukemia samples and looking at a variety of different genomic effects in there. And the take-home message from that is structural variation and complex rearrangements and other variations are critically important in comprehensive understanding of pediatric cancer. And this also extends through to even other conditions such as sickle cell anemia in the characterization of the HBG1 and 2 locus, where those 2 loci share 90% identity with each other. And so there are key SNPs that define sickle cell phenotypes that exist in one of the genes and not the other, and being able to resolve them considering that identity is of critical importance. They're very robustly resolved with long read approaches that can read across the regions of identity and into regions of uniqueness, and then phase and appropriately characterize the variants, whereas traditional short read sequencing struggles with those areas. And then finally, conditions like Ewing sarcoma and chromothripsis, where you have these very complex, almost chromosome shattering sort of events, are really only well resolved with longer read approaches. And so with some of that background and recognition of some of my colleagues that are contributing to that, I'm going to move fairly quickly through a variety of data sets and some observations that have been made. But I also wanted to provide a very brief introduction. I won't read through all the text that's shown on the slide. As was mentioned, this will be recorded and available, so it gives an opportunity for someone to go back and see that. But basically, part of the All of Us long read sequencing project is a goal to sequence between 3,000 and 6,500 samples to complement the array and short read sequencing data. We want to make a population scale set, or the beginnings of a population scale set, using long read approaches to really evaluate the structural variation that is present in the All of Us research program participant cohort, and really determine population frequencies for indel and structural variants to improve calling of SV events, really over all data types. So not just long read sequencing, but leverage the long read sequencing to improve analysis in short read and vice versa. And we also want to improve resolution in challenging regions of the genome, such as pharmacogenomic genes, the HBG1 and 2 locus that I mentioned, other regions such as SMN1 and 2, and a variety of medically relevant regions. So I'll mention some specific work on that in a few moments. And so we've been able to validate the approach pretty well. This next section will kind of walk through some of that, and I think what should hopefully be some very interesting observations in the amount of long read sequencing necessary to comprehensively characterize a human genome. So I think this probably comes as no surprise. What you're looking at here is simply that circular consensus sequencing, or HiFi reads, are better at resolving complex regions than short reads. So you see HiFi reads are just shown at the top via an IGV view, and then corresponding reads from a short read approach are shown on the bottom. And so you can just see, for this particular region, much better resolution and overall performance in terms of variant characterization in the top versus the bottom. So again, probably very little surprise there. So when we look at a short read versus long read genome. And I'll show you some data to support essentially what this summary shows on this particular slide in a moment. Is that we kind of have two ends of a spectrum. On short read genomes, we know that there is an exceptional value in terms of cost per genome or cost per base, and that the high per base accuracy and consensus accuracy allows very, very strong sensitivity to single nucleotide variants. And I think that was, certainly been borne out by the literally millions of genomes that have been sequenced worldwide. However, detection of complex variants and resolution of structural variants that are significantly larger than the read length remains a challenge. And I think the field has come to an understanding that 30x genome coverage remains a standard for genome sequencing approaches. So through some of this early work with the All of Us research program, as well as involving some fantastic scientists in a few different consortiums, including the Telomere to Telomere Consortium, the Human Genome Structural Variant Consortium, and some others, there's now options of bringing scalable long read approaches forward. Now, certainly there needs to be continued development to bring costs and throughput into the same realm as short read sequencing. But again, it's time. I think it's time to robustly look at doing population scale sequencing. Hence the funding that we were fortunate to get for the All of Us program to support this. So the punchline is shown on the bottom right side of the slide, is that a 10 to 15x HiFi coverage is an emerging option that balances cost and performance. And what I'll show you in the next couple of slides is a very simple conclusion, and that is a 8 to 12x HiFi genome has the same variant calling performance as a 30x short read genome. And in fact, that's starting to be a metric that we're bringing forward on these population scale studies. So it's really I would say, stated differently, the idea of needing to go to a 30 or 40x HiFi genome is certainly a great resolution to have in that data. And if all things were considered equal in terms of cost and throughput, no one's going to turn away that opportunity for that amount of data. But what the next few slides will show is that it's that amount of data is not necessary to achieve a 30x short read genome like performance. So before getting there, just a couple quick slides on through work by Nirpesh Prasad and others in the HudsonAlpha Discovery group. There's been a lot of optimization work around HiFi library generation, essentially going now with a typical input material of 3 to 6 micrograms of material as far as optimal, and then using some preparative changes and opportunities to scale. We're now routinely producing libraries with a 20 kb insert, and so for really all of the data that you'll see and the conclusions that you'll see from that data were libraries of this type. So they're HiFi sequencing, generally 1 to 2 smart cells per sample, with the vast majority of the data being 1 smart cell per sample. So in that 8 to 12x coverage, depending on the yield of the smart cell. And then again, insert sizes in the 18 to 20 kb range is where they've been targeted. So what that's allowed us to do then is using 2 laboratory technicians and 2 megaruptor instruments to shear the DNA. It's a 32 samples per week, 96 samples per batch workflow. So essentially, library preparation and size selection for 96 samples is completed over the course of 3 days or so. Polymerase binding, of course, happens at the start of the sequencer. So what we end up with is a preparation cycle of 5 days, and then the sequel 2 started on the 5th day. And so you kind of see the cartoon version on the bottom, where collection 1, 2, and 3 as we go, followed by immediate analysis on the data processing side. So what this allows now is with 4 SQL instruments dedicated and running essentially nonstop, you can get through about 96 samples a month using a single smart cell per sample. And so that's kind of the workflows that we are moving forward at this time. And importantly, we also see there's no significant differences in performance between sequencing instruments. This is something we looked at. And so again, I won't read through all the numbers, but if you look at instruments which are bracketed in red on the left versus batch, which is bracketed in dark color blue or black on the right hand side, depending on how it appears on your screen, you can see reasonably consistent insert sizes and very consistent polymerase read yields. And the variance you see there is what we typically see in some of this variance. We've gotten a little bit tighter range now in some of the later batches on the insert sizes, but the polymerase read yields are pretty typical. And then you can kind of see some of the other metrics in terms of raw versus unique molecular versus CCS coverages. So and I guess these next 3 slides are really the punchline for my conclusion that you can get away with lower coverage on PacBio. So what you're looking at here, and there's 3 slides that will look very similar. One for structural variants, which is shown now, another for indels, Structural variants being 50 nucleotides and larger in size, indels being from 1 to 49 nucleotides, insertion deletion events, and then finally SNVs or single nucleotide variants. All the slides are structured the same way. This all was analysis done by Pete Adano at Evan Eichler's group using data produced at HudsonAlpha Discovery. And so what this is a measurement of how data improves by adding incremental smart cells. So everything listed under set 1 is 1 smart cell, set 2 is 2 smart cells, 3 and 4 are 3 smart cells and 4 smart cells respectively. And so you see kind of a graphic on the right-hand side of the slide and then specific numbers indicating the precision, recall, and F1 rates on the left-hand side of the slide, with and without terminal repeats, is the all versus no TR. So with that introduction, I'll go through the next couple of slides pretty quickly, but hopefully what you can see here is note that there's very little change in false negative, true positive, or false positive rates as you go from 1 smart cell indicated in column 1 on the right-hand side of the slide versus 4 smart cells. So by quadrupling your data cost, quadrupling your overall amount of data per sample, you can see your recall only increases a very small amount, precision increases a very small amount. And again, this is for structural variants. So a single smart cell per genome for structural variants clearly does basically as well of a job as 4 smart cells. If we ask the same question for indels, we see a mild improvement, which isn't too much surprising as you go from perhaps 1 smart cell to 2, but then very, very small improvements as you go beyond 2 smart cells per sample. And then finally with SNVs, the most significant improvement is in decrease in false negative rate, again, as you go from 1 smart cell to 2, but then essentially 0 benefit of going beyond 2 smart cells per sample. And this was all done on genome in a bottle type samples, analyzed a variety of different ways, and I'll show you some of that data in a moment. But punchline, again, hopefully in support of that conclusion that a 8 to 15x HiFi genome has exceptional performance and as good a performance as a very deep short read genome. So if we do ask the question and then broaden the comparison between Illumina sequencing, Oxford Nanopore Promethean sequencing, and then HiFi sequencing, and ask, I think a fairly simple question with a somewhat complicated answer, as you can see in this graph. And again, this is additional work done by Pete Adano. If you look at sensitivity versus indel size, the one area that the HiFi sequencing really does stand out dramatically from the other approaches is in those medium range indels. So HiFi captures indels much more consistently than any other technology. So the solid lines you see at the top are the HiFi data. The dotted lines in the middle or the small dotted lines in the middle is Illumina, and then Oxford shown on the bottom. And then you can see the sensitivity improvement. So exceptionally improved sensitivity for these indel events. So if we are asking the question, at least as we are in all of us, which is what is the methodology that allows us to capture all 3 variant classes in the most robust manner and with the highest accuracy, HiFi sequencing has certainly stood out. So if we then extend that to a slightly different question, and so many of you are probably familiar with this publication that came out in 2016, which in a nutshell was an effort to look at highly homologous genes with clinical relevance and start developing resources for clinical next generation sequencing considering these things. So being that this is about a 5-year-old effort or so, this predates any discussions of routine use of long read sequencing. But we took the 193 gene list from this group and then started asking how well does it resolve within the data sets that I've been describing so far, meaning does this modest coverage, that 8 to 12 to 15x coverage via long read sequencing or HiFi sequencing allow resolution in these 193 genes that are quite challenging for a few different reasons. So this work was primarily done by Medhat Mahmood and Fritz Sedlizak at Baylor, again as part of that long read group. So this is a subset of samples. In this case, 18 HiFi samples. There were two samples in a set of 20 that were done as part of a preliminary part of this study that had very low coverage, and so at the time we did this analysis, they were removed. But I wanted to show this slide simply to indicate that in this particular cohort, and this was done intentionally, that we wanted to see the effect of a range of coverages. So as high as 22x via HiFi sequencing, and then a low of just over 5x. Actually, I should say almost 5x. So a pretty good range of coverages, and we wanted to really assess what the minimum depth of sequencing would be necessary for a passing metric as part of the All of Us program as it related to clinically relevant genes. So if we look at the mapability, the point here, so it's genes along the bottom and then mapability. All I wanted to point out with this slide is note that the red shows version 37 of the genome, and then the green shows 38. This just clearly indicates that your choice of reference is of critical importance as you go through whatever the goals of the analysis, and I'll touch on that again later. But again, so just the point being that mapability was good, but choice of reference is important. If we asked the question, how many genes are zero covered, there were a collection. Fortunately, the vast majority of those genes that had ended up uncovered in some samples, they were Y chromosome genes. So obviously in any female samples, those would be uncovered. And then outside of the sex chromosome genes, there was a relatively small number of genes that remained uncovered in this exercise with that low coverage. And we did see that if we increase coverage, you gain back much of this, or I should say, number of genes dropped from 15 to 12 by increasing coverage. So we gained a few genes back. So in this case here, but there are some additional benefits. If we asked what percentage for the read lengths being long across these HiFi samples over the 193 genes, it asked SNV calls, heterozygous calls, and then what percentage of genes we could phase. This is certainly another thing that the long-read sequencing brought to the table, is the ability to phase variants across these genes, which is also something that was not readily resolvable with other methods. So not only are the genes being well resolved, but we're also able to phase variants to a pretty high degree in the high 70s to low 80% range with the coverages that we've talked about and the insert sizes that I mentioned previously. So if we look at the phase blocks per gene, as you see, this is sort of just slightly different view. And instead of looking at percentage, it's actually looking at number of genes phased. So they're, again, in the 160 range or so, as you see across the different samples. Not a lot of dramatic differences in phasing of the variants based on coverage, which was a good result. And then for some of the samples where the phasing wasn't quite as complete, you can see the increase in phase blocks per gene shown on the right-hand side of the slide. And so then if we finally look at a truth set versus structural variant summary, so this is stepping away from not only just single nucleotide variants, as we look at structural variant length and then structural variant type. So the type is color-coded, as you can see on the far right-hand side of the slide. And then the length is shown on as SV length on the axis, and then the count is that we were able to resolve about 25,000 structural variants, with majority of those being insertions and the minority being deletions, with a structural variant length of greater than 50 in the same data set. So if we now, this is where things get both complex as well as interesting. And there's today's seminar isn't quite enough time to delve into this in a tremendous amount of detail. So the point of these next couple of slides is indicating that not only is your choice of reference important, but your choice of tools are important. So what you see in this case, again, this is also work that was done in collaboration with Baylor. And you're looking at representations across the truth set that was mentioned in the previous slide, but now comparing by tool. So you have Sniffles, PBSV. The truth set is the truth set from Genome in a Bottle, QtSV, and then SVIM. So all of those different tools were run on the data sets, and then MedHap performed a review of asking where we saw divergence or convergence of the different results, and that's what you see in the lower part of the slide. So on the far right-hand side, the tallest blue bar is where there was consensus across all of the tools as well as the truth set. So the vast majority of variants, a little over 12,000, was concordant regardless of which tool was used and with the truth set. And then if you look on the far left-hand side of the slide, you can see the collection of variants which were unique to any one of those populations, whether the truth set, so there's about 3,000 variants that went undetected with any tool in this data set. We're still investigating some of those reasons, and then you can see each tool had a subset of variants that was specific to those tools, and then it was just the combinatorials all the way across. So you can kind of see where, for example, there was another 3,000 variants or so that everything but sniffles was able to capture. And that was so meaning that it was in PBSV, TruthSet, QtSV, and SVIM. You see that as the tall blue bar, as the tall black bar towards the right-hand side of the slide. So the point being is that regardless of tools, we captured the majority, and then it's really a refinement of the tools and optimizations of the tools to go through the final polishing of the data of sorts. So if we then ask, what is the precision and recall for the different permutations? And what stands out here, and this, again, so this was hopefully a reasonably simple way of looking at a complex set of data. So slightly different view than what you saw on the previous slide, but now this is kind of the convergence of a computer science way of looking at this with a way of making a figure. So you see the same tools listed across: QtSV, PBSV, Sniffles, SVIM, PATH tools, and TruthSet. And now asking, now where you see the ones, the dots versus the ones are which of the combinatorials. And the key thing to show is that in this particular case, the best balance of precision and recall was with PBSV compared to the TruthSet, which is shown about a third of the way over from the left, where you see the precision and recall are quite close. Everything else is you can see better precision or a sacrifice in recall, or better recall, a sacrifice in precision, but that best balance does appear to be with PBSV versus the TruthSet, and we're continuing to look at these things pretty carefully. But the bottom line is tool choice, et cetera, is quite important in these studies. And then in this particular slide, again, it's slightly different view with, again, the PATH tools. In this case, calling single nucleotide variants rather than structural variants. And in this case, the tools that were used were PATH tools, TruthSet, long shot, or deep variant. And again, vast majority called by all four, large number of variants uniquely called by deep variant. We're looking into that now. This is most likely a training set, we are very confident deep variant will get quite a bit better with the long read data as the training sets get more comprehensive. So we're quite enthusiastic about that. But if we look at some of the other ideas, PATH tools, long shot, and deep variant do seem to detect a number of variants unique. And then if we leave out PATH tools and just use the TruthSet detected with TruthSet, long shot, and deep variant, you see we capture back another significant proportion of variants that were missed. Or we capture back the majority of the TruthSet variants that were missed across all four together. So again, point being, choice of tools, very important. So I'm going to shift gears, and I'll kind of touch on some of the conclusions from this previous segment, but shift gears a little bit into work that was done by Nipresh Prasad in our group in collaboration with, or in support of a clinical trial from Wave Life Sciences. So Wave has publicly disclosed all of this information in the past. And the idea is an extension of what was previously presented around the COVID work, where if you can produce a long enough amplicon and then sequence across that amplicon, you have resolution for phasing and resolution for characterization that would be impossible with other methods. And that's exactly what Wave took advantage of in characterization of expanded Huntington repeat regions. So this is a CAG repeat expansion, where if you have 36 or more copies of the CAG repeat, which expands a glutamine track, you end up with pathology of the disease. But there are two variants that are quite important for potentially targeting the expanded versus the wild type allele that are in phase with the expanded variant, or say the expanded allele. And so there was a requirement from Wave of being able to capture, and I think the next slide should have a better, no, so I thought I had a better cartoon of it. But if you look in the, let me see if I can, I wasn't finding the pointer, but that's okay. So note where it says expanded CAG 36, and then you see the SNP shown on the middle, just above the cartoon of the brain. So in this case, there's a long amplicon that's produced. This is done off of an RNA transcript that's converted into cDNA, that's then converted into a sequencing library, where we then characterize the presence of a T SNP shown in the middle of the slide versus a C SNP with the expanded allele versus the single. And this is how Wave characterized samples that are eligible for really an amazing nucleic acid targeting drug that uses a very pure stereoisomer, which is of a single form. So if there is the correct SNP that's in phase with the expanded allele, they can specifically target only the expanded allele in the Huntington's patients, while leaving the wild type allele intact, or I say unaffected. And that's really the significant advancement that this trial has brought forward, is the ability to make that change. So, you know, a pretty amazing result. And this this slide describes the pre-screening and screening process. Essentially, it's that the patients have the correct SNPs, they have an expanded allele, and then if both of those are true, then the patients are admitted or are allowed to proceed into the phasing study, which takes advantage of the HiFi data. And then here we can see the concordance of CAG sizing data from known standards, and these violin plots just indicate, as you can see, where they're clear separation, relatively low noise in the overall set, that it's an exceptionally performing assay. Meaning that the patients that go through this and the samples that have gone through this have validated very well. It's been a very, very successful effort, and so far a very impactful trial in Huntington's disease so far, by being able to specifically target only the expanded allele and do so in a manner that leaves the unaffected allele intact. So, and lastly, I just wanted to touch on some of the analysis complexities, and this is work that I mentioned from Kieran Garamella in the last couple of slides, and I'll move through these pretty quickly, but this is in support of some of the earlier comments that choice of tool and choice of reference is critically important. And I'll sort of give you the conclusion before the data. And the conclusion is, for the All of Us research program, we are now Kieran and colleagues have largely developed a analysis pipeline that takes advantage of the Sequel IIe CCS processing local to the instrument, followed by cloud analysis in the Google Compute Cloud platform, to allow us to use multiple tools, actually a very comprehensive set of tools that includes most of what you saw on these earlier slides, and of critical importance, multiple references. So we're not only aligning to just GRCH38 or the HD19 type references, but we're also including the hydidiform mole reference, CHM13. And the reason that's important is there's a large number of structural variants, or I should say there's a meaningful number of structural variants that are not well annotated in the traditional references that are well annotated in these, and call them they're more assembly-based metrics. And then finally, and again, I'll illustrate, kind of giving you the conclusions before the data, but we are also running assembly-based analysis methods as part of this pipeline, because there's additional resolution that the long-read data brings forward that some of the other tools don't detect. So this probably paints a fairly complex analysis picture, and that's true. But we're very excited with what it'll reveal in the thousands of sample range over the next year or so that we're doing for the All of Us program, and then also equally excited this analysis will happen in a very scalable cloud platform. And so again, this is the next several slides. You'll see work from Kieran and colleagues at the Data Sciences platform at the Broad Institute, and really just a phenomenal amount of work here. And the key thing is that almost all of these analysis platform, or all of these analysis steps, have been set up in a way to use preemptable compute nodes, or which allows access to preemptable pricing, which essentially is about a 75% discount in the exchange for the cloud platform to have the right to preempt and reschedule a compute job. And so the entire analysis platform was set up kind of around that. So you can do the same configuration in an AWS, and this gives you an idea that you go from about $2 an hour to about 56 cents an hour for the same node. In order to make this work, to capitalize on those preemptable compute discounts, all of the subreads are basically shattered, or what Kieran refers to as they shard the subreads and then dispatch about 300 jobs on modest hardware, each running for about an hour or two. Because the preemptable nodes, generally, if your job only takes an hour or two, it'll almost never be preempted. It's only if you go to the extended times. And so this is a very, very effective way of doing this. And then you can potentially further future savings with memory increasing retries and resumption from checkpoints in the preemptable task, meaning there's methods that are underway now that if a job gets to a certain checkpoint, instead of restarting the entire job, you just start from that checkpoint forward if it happens to get preempted. So the starting strategy really also allows other tasks to be run on the preemptive nodes, and this is just a cartoon of that here, meaning that you can also do the same thing if you were to perform the CCS analysis via the cloud. Now, the decision point that we made versus the 2E upgrade versus doing CCS analysis in the cloud was very simple, and it had mostly to do with storage management and the amount of data that had to be moved. So we did by doing some of the calculations. It'll be long-term far more efficient to allow the CCS analysis to happen on the compute nodes local to the sequencers, and then move about 1/3 of the data we otherwise would have to the cloud and start post CCS analysis in the cloud. But the concepts and what Kieran and colleagues have proven out is the CCS analysis is equally efficient in the cloud, as well as some of these other techniques. So it is a viable option. So total cost for processing 24 samples, including CCS correction. So again, in this case, this was pre-2E upgrades in our case, including CCS correction, flow cell merging, assembly, SNV and SV calling by a multitude of tools, basically comes down to about $61 a sample. And so it's a pretty, very cost-effective, very cost-efficient way of doing these things in a multiple sample basis. And then finally, you can also manage the overall compute cost by migrating data to lower tier storage units over time using buckets with special retention rules. This is also something that's been worked on. And what you see here shown on the left-hand side of the slide is basically everything moves down a tier after the first 30 days and then after 120 days. And then you can see the aggregate storage costs for 600 gigabases versus 200 gigabases, with 1 month in standard storage, 3 months in nearline storage, and then 8 months in cold line storage. So storage costs for one flow cell is shown below, whether it be at a 600 GB or 200 GB yield. So it's about $30 versus $10. So again, punchline being creative and efficient with some of the available cloud computing resources can lead to a very manageable both storage and compute costs for this type of data. And then finally, there is a high degree of annotation, a high degree of programmatically accessible information, including per flow cell and per sample file paths and metrics that are automatically appended to the data model. So again, they're programmatically accessible, which is critically important for large scale work. And then importantly, so we did have an opportunity to ask a simple question. If we perform these analyses in a very traditional manner, using local compute nodes, cluster computing, et cetera, and then did the processing, the cloud-based processing I was discussing a moment ago that's based in Terra, and asked, do we see the same answer? And the answer is yes, that we see very broadly consistent metrics between what was done locally at HudsonAlpha, what was done in the cloud, and these comparisons have been done a few different ways. So what this means is the cloud-enabled tools are creating the same answers and the same data sets as things done on local compute. So this was an important result. So lastly, and I think this was alluding to what I mentioned earlier, within this platform, we now have the ability to do HiFi ASM assemblies, GRCH38, as well as CHM13 based alignments and variant calls, all within the same pipeline. So we're analyzing the same data sets and able to do these kind of, call them a new resolution in whole genome sequencing to get the greatest performance from, or extract the greatest value from the long read data and get the resolutions that are possible with these data sets that otherwise went undistinguishable. So, I guess I want to make sure I don't think I can overstate the value in this and the and how excited we are to bring this long read platform forward in collaboration with such a great group of scientists. Again, particularly because we're now doing alignment-based as well as assembly-based analyses, and then bringing all this together to a truly unprecedented level of analysis of the human genome that is all underpinned, or the foundation of all of that is the HiFi sequencing. The accuracy of the HiFi sequencing and the length of the reads is really what's driving nearly all of this. So that's the end of the presentation I had. So thank you for your attention. I know we went through a lot of data very quickly. Always happy to take questions, whether it's in the chat form here or feel free to reach out to me afterwards.
Thank you, Shawn. That was an excellent presentation. And I will invite all participants to submit questions in the chat box. We do have a couple of questions that have come in. So, Shawn, the first question I think I'll direct to you. Did HudsonAlpha consider Bionano Genomics Sapphire system? Yeah, absolutely. So Bionano is a fantastic platform. The resolution that it brings is, sort of,
I would say is another level of capability. Our only holdup, and again, our biggest consideration in BioNano was two factors. One is throughput, and the other was cost per sample. But as far as bringing value to these exercises and again developing high resolution analysis of genomes, et cetera, I'm a huge fan of the BioNano platform. Just and continue to keep up to date with those folks. I mean, it's a great group there. Love the platform. It's just something that just didn't fit into our both budget and throughput models. That was our really bottom line.
Great, thank you. Last question, I think, for given the time constraints, and I think, Cheryl, this one is for you. What type of coverage is required for targeted sequencing applications?
Hi, thanks, Lori. Yeah, that's a great question, and the answer is pretty complicated. It all depends on what your sample is, what your goal is, and so forth. So in terms of if a targeted application, if you're looking at heterozygous sample and a reasonably high multiplex level, you'd like to make sure that you get good coverage of both alleles for all of the samples that you're multiplexing. Then our kind of general rule of thumb there is something like 50x coverage, 50-fold coverage per allele. That allows a lot of leeway for allelic imbalance in your targeting, as well as variation in yield from a number of different samples.
Great, thank you. All right, well, it looks like we are coming up to the top of the hour, so I want to remind everybody and thank everybody for attending that the session has been recorded, and we will be making that available to you. And we welcome your feedback. There is a brief attendee survey that you can link to through the QR code shown on the screen. And of course, please feel free to contact Discovery Life Sciences at dls.com/PacBio webinar, or contact a PacBio scientist at pacb.com/scientist. And with that, I think that we are going to conclude our webinar for the day. And again, thank you all for attending, and thank you, Shawn and Cheryl, for your time and terrific presentations.